Hey there, have you ever stared at a genetics problem and felt like you were just guessing based on patterns instead of really truly getting it? Well, today, we're going to change that. We're not just looking for the right answers, we're taking apart the questions themselves from a real university-level genetics test. We're going to uncover the logic, step by step, so you can see genetics as this incredible puzzle, not just a bunch of rules you have to memorize. Let's dive in. So, here's our game plan. We're going to kick things off with Gregor Mendel's two big laws. I mean, this is the absolute foundation of genetics. Then, we'll build a bridge from those abstract ideas to the real world by actually locating these genes on chromosomes. After that, we'll jump into the world of data with the chi-square test, which is how we figure out if a genetic theory holds up. And to wrap it all up, we'll explore some of the really cool advanced stuff, the exceptions and complex interactions that go way beyond the basics. It's a full journey, from core principles to real practice. All right, let's get into Section 1, Mendel's first law. So the question we're breaking down is this. State Mendel's first law and use it to explain the outcome of a cross between a homozygous yellow-petaled tomato plant and a homozygous red-petaled tomato plant through to the F2 generation. Okay, so what is this question really asking? It's testing two things. First, can you just state the definition? And second, can you actually use that definition to predict what happens to the offspring for two whole generations? The key words you really need to zero in on are homozygous, cross, and F2 generation.
So the first part of the answer is just knowing the name. Mendel's first law is called the law of segregation. And the core idea here, and this is totally crucial for solving problems, is that for any trait, like petal color, an organism has two versions of a gene, which we call alleles. Now, when that organism goes to make a sperm or egg cell, those two alleles don't blend. They literally segregate or separate from each other. That means each sperm or egg gets only one of the two copies. This clean split is the engine that makes all of Mendelian genetics work.
Okay, now let's actually apply this law. The problem says we have a homozygous yellow plant. Homo means same, so both of its alleles for petal color are identical. We'll write that as big Y, big Y. It's crossed with a homozygous red plant, so little y, little y. Because of segregation, that yellow parent can only make gametes that have a big Y, and the red parent can only make gametes with a little y. So when they cross, every single baby plant in this first generation, the F1, is guaranteed to have the genotype big Y, little y. And what do they look like? Well, they're all yellow because the yellow allele is dominant. It looks like the red trait just vanished, but really it's just hiding in the background. So how do we get that hidden red trait to show up again? We take two of those F1 plants and cross them. This is where the Punnett square becomes your best friend. Look at the top and the side of the square. Because of segregation, each of those YY parents makes two kinds of gametes. Half get a big Y and half get a little y. The square is just a simple way to map out every possible combination. If a big Y gamete from one parent meets a big Y from the other, you get YY in that top left box. If a big Y meets a little y, you get YY. You fill out the whole thing and look at what you get. Genetically, it's 1 YY, 2 YY, and 1 YY. But phenotypically, what they actually look like, the YY and the YY plants are all yellow. So that's 3 yellow plants. And only the YY plant is red. And there it is. That classic 3-to-1 ratio. The law of segregation predicted it perfectly. Okay, so we've nailed how a single trait gets passed down. Now question 1B levels up the difficulty. What happens if we're tracking two traits at the same time, like petal color and seed color? To do that, we need another tool from our toolbox, Mendel's second law. The question asks us to state the law and then explain the gametes that a YYBB parent would make.
This law has a fancy name, the law of independent assortment. But all it really means is that the gene for petal color and the gene for seed color are inherited completely separately from each other, as long as they're on different chromosomes. They don't travel as a package deal. The allele you get for yellow petals has absolutely no effect on whether you get the allele for black seeds. They're independent events. It's just like flipping two separate coins at the same time. This idea is the whole key to predicting outcomes for more than one trait.
Now this slide here, this is probably the most critical step in any two-trait problem, and it's honestly where most mistakes are made. We've got a parent who is heterozygous for both genes, YY and BB. Independent assortment tells us that how the Y alleles get sorted has zero effect on how the B alleles get sorted. So that big Y allele has a 50-50 shot of ending up in a gamete with the big B or with the little b. Same thing goes for the little y. This gives us 4 and only 4 possible combinations for the gametes, and they're all equally likely. YB, 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 and YB. If you can master this step, you can solve any dihybrid cross you see. Okay, now picture a much bigger Punnett square, a 4x4 grid. Along the top, you'd list those 4 possible gametes from one parent, YB, 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 YB. Then you do the exact same thing down the side for the other parent. That gives you a grid with 16 boxes. Now, you don't always have to draw it all out. If you did, and then you counted up what all the offspring look like, you'd get this absolutely iconic result you see here, the 9:3 to 3:1 ratio. You get 9 plants showing both dominant traits, 3 showing the first dominant and second recessive, 3 showing the opposite, and only 1 lonely plant showing both recessive traits. If you see this ratio in a genetics problem, a light bulb should go off in your head that says dihybrid cross. Okay, so we've been throwing around these letters, these alleles, like they're just abstract concepts. But where are they physically? Question 1C makes us connect the dots, asking us to actually draw them on their chromosomes. It's about linking the abstract genetic model to the reality inside a cell. It even tells us the petal color gene is on chromosome 4, and the seed color gene is on chromosome 20. First things first, we need to know how to draw these chromosomes the right way. The question tells us their structure. Chromosome 4 is acrocentric. That just means the centromere, that pinched-in part, is way off to one side, so you get one really long arm and one tiny little arm. Chromosome 20 is metacentric. Meta sounds like middle, and that's exactly where the centromere is, creating 2 arms of about equal length. These aren't just random details. You need them to draw an accurate diagram. So now, let's put our letters on these drawings. For homozygous individual, say big Y, we draw a pair of those lopsided acrocentric chromosomes, and we put a capital Y allele on the exact same spot, we call that the locus, on both of them. For a heterozygote, Y, small y, the drawing is the same, but one chromosome gets the big Y allele, and its partner gets the little y allele, right at that same spot. And we do the same thing for the B and B alleles on those balanced metacentric chromosomes. This is it. This is the visual link between Mendel's abstract laws and the physical stuff inside our cells. All right, time to switch gears. Let's move from inheritance patterns to a little bit of statistics. Question 2 gives us some data from an experiment on a plant disease treatment and asks a pretty straightforward question. Is there a real, statistically significant link between the treatment and the outcome, or did these results just happen by random chance? To answer that, we need a really powerful tool called the chi-square test. The very first step for any chi-square test is just to get your data organized. The problem gives you a bunch of numbers. 10 treated plants had symptoms, 10 didn't, and so on. We take those observed numbers and arrange them neatly into what's called a contingency table, just like you see here. It's basically a grid. On one axis, we have treatment status, and on the other, symptom status. The table doesn't just hold our data, it makes it super easy to calculate the totals for each row and column, and we're going to need those for the next step. Now, before we crunch a single number, we have to formally state our hypotheses. Think of it as setting up 2 competing ideas. The null hypothesis, or H0, is always the boring one. It's the default assumption that there's nothing interesting going on. In this case, it says there's no relationship at all between the treatment and the symptoms. The alternative hypothesis, H1, is the opposite. It's the one we're actually curious about, that there is a connection. The whole point of the test is to see if our data is strong enough to let us reject that boring null hypothesis. Okay, this formula can look a little scary, but let's break it down together. We'll just do the calculation for one box, the top left one for treated symptoms. Step 1, we have to figure out the expected value. The formula is simply row total times column total divided by the grand total. So for that box, it's 20 times 16 divided by 50, which gives us 6.4. That's the number of plants we'd expect to see in that box if it was all just random chance. Step 2, now we compare what we expected, 6.4, to what we actually saw, 10. The formula is observed minus expected squared, then divided by expected. So that's 10 minus 6.4 squared, which is 12.96, divided by 6.4, which gives us 2.025. Step 3, we would do that exact same calculation for the other 3 boxes in our table, and then just add up all 4 of those results to get our final chi-square value. So after we do that calculation for all 4 cells and add everything up, we get this number, 4.963. This single value is our chi-square statistic. You can think of it as a measurement of how much our real data deviates from the boring world where there's no connection. So the big question is this number big enough to be meaningful? This is the moment of truth. To make a decision, we compare our calculated value to a critical value that we get from a statistics table.
The problem gives it to us, it's 3.841. Now, the rule is incredibly simple. If our calculated number is bigger than the critical number, our result is statistically significant. And that means we can reject the null hypothesis. Since our 4.963 is definitely bigger than 3.841, we can confidently say that yes, there is a statistically significant association between the treatment and the symptoms. It's very unlikely we got these results just by a random fluke. For our final section, we're moving beyond the straightforward rules of Mendelian genetics. Question 3 is basically a vocabulary test, asking us to tell the difference between several pairs of more complex genetic terms. This is really testing our grasp of the nuances and exceptions to the rules, which is where genetics gets really, really interesting. First up, we have codominance versus overdominance. This is all about how two different alleles behave in a heterozygote. With codominance, they're both fully expressed. The classic example is AB blood type. You aren't a mix of A and B, you are fully 100% both at the same time. With overdominance, which is also called heterozygote advantage, the heterozygote's phenotype is actually more extreme or provides a bigger advantage than either of the homozygotes. The textbook case is sickle cell trait, where being heterozygous makes you more resistant to malaria. So, an easy way to remember, codominance is both, overdominance is better. Next, let's compare an inhibitor gene to a lethal gene. The key difference here is the consequence. An inhibitor gene is a type of epistasis. That's when a gene at one location can mask or block the effect of a totally different gene. Think of a gene for no pigment production, which would hide a gene for black fur, making an animal white. A lethal gene, on the other hand, is way more dramatic. If an organism inherits a certain combination of these alleles, it doesn't survive, often dying before it's even born. This really messes up the expected Mendelian ratios you see in the living offspring. So one affects a trait, the other affects life itself. With this pair, the difference comes down to the number of genes versus the number of alleles. Supplementary genes are when two different genes work together to produce a new trait. For example, maybe one gene makes a red flower pigment, and a second gene has an enzyme that modifies that pigment to make it purple. It's a team effort. A polymorphic gene, on the other hand, is just a single gene that happens to have more than two versions or alleles floating around in the population. The ABO blood group is a perfect example with its A, B, and O alleles.
And for our last pair, we've got duplicate genes versus pleiotropy. You can think of these as complete opposites. Duplicate genes are when you have several genes that all do the same job. It's a many genes, one trait situation. It's like having backup systems. Pleiotropy is the exact reverse. It's when one single gene has an effect on multiple, often seemingly unrelated traits. For example, the gene that causes Marfan syndrome affects the skeleton, the heart, and the eyes. So just remember, duplicate is many genes, one trait, and pleiotropy is one gene, many traits.
And that brings us to our final thought for the day. Pleiotropy isn't just a term to memorize for a test. It's a huge real-world challenge in medicine. If a single faulty gene can set off a whole cascade of different problems throughout the body, it makes everything from diagnosis to treatment so much more complicated. It really forces us to move past that simple one gene, one disease idea and start to appreciate the incredibly complex, interconnected network that is our own biology. Thanks for digging into genetics with me today.